By for my talk. All right, so the origins and establishment of an allopolyploid and the consequences for geographic range. So just to quickly sort of overview my research. So I think sort of as evolutionary biologists, we can all kind of appreciate the diversity of forms that we encounter in nature. Um, and we have developed sort of lots of explanation for this diversity. However, I think I would argue that sort of documenting the causes and consequences of interspecific variation can really help us to sort of understand the changes that take place at the individual or population scale, which sort of influence the evolutionary trajectory of species. And sort of variation in genome structure is one common kind of interspecific variation in flowering plants, but how it sort of links with other kinds of variations, such as phenotypes and fitness, is often unclear. And I'm sort of also interested in how changes in genome structure relate to sort of broader phenomena, such as geographic range size and speciation. So focusing in on one of these particular kinds of variations, polyploidy, I kind of am interested in polyploidy because of its role in speciation in particular, but polyploidy is kind of common in terms of how it influences traits in an agricultural setting, and also its sort of implications in the origin of angiosperms writ large. But it's also, like I said, a very common form of speciation. And it's really interesting also because speciation in polyploids comes with some special challenges that are quite different than speciation as we sort of normally think about it. So what are these challenges and how do they work? So first, you have to understand sort of that there's kind of two broadly kinds of polyploids that we can find in nature. Those are called autopolyploids and allopolyploids. Allopolyploids essentially are when you have whole genome duplication accompanying hybridization, whereas autopolyploidy is sort of just within a single genome. And when polyploids are first formed, they have a high degree of a really high degree of post-zygotic reproductive isolation because of this increase in genome size and genome copy number. That means that they cannot form fertile offspring with diploid relatives. So in the case of sort of diploid speciation, you might expect that post-zygotic reproductive isolation will increase over time, over some sort of timescale, whereas when you have speciation via polyploidy, you get this immediate jump in post-zygotic reproductive isolation in sort of a single generation, which is a really profound and sort of dramatic change that we don't typically see. So kind of moving beyond speciation, following the origin of a new species via polyploidy, there's kind of these common trends that are thought to sort of occur in polyploids. Those two trends are often geographic range size and niche shifts, with polyploids often being thought to sort of have larger geographic ranges and having different, sometimes larger abiotic ecological niches. And sort of we want to understand the role of polyploidy in these shifts, or if these shifts are really even taking place. But sort of the role of polyploidy in these phenotypic and ecological shifts are actually kind of tricky to figure out. So kind of fundamentally, we don't really know whether changes in range or niche are driven directly by polyploidy exactly, because they could have occurred potentially in a non-polyploid taxon or were the result of evolution sort of after the polyploid formed. So if you kind of look through this diagram here, a genome sort of doubles, it takes up more space in the cell. This can cause alterations to the physiology of the plant because of increased cell size. And this is presumed to have some sort of effect on the niche sort of instantaneously. So we sometimes call that like polyploidy per se, or the immediate impacts of polyploidy via cell size. And as I sort of mentioned, this effect of polyploidy per se could be large in terms of how it influences phenotypes or ecological trends or patterns. or it could be relatively small compared to the later sort of evolution that's taken place after the polyploid has formed. And it's sort of this gap here is whether or not, this effect of polyploidy per se is really what's driving observed shifts in polyploid ecology. So sort of the overall guiding question here is, how does polyploidy per se contribute to the establishment, persistence, and range shifts of allopolyploid plants? I should mention also that the reason that we're focusing on allopolyploids here is because even though allopolyploids form far less frequently than autopolyploids do in nature, they're at least equally as common, suggesting that there's a really strong advantage, like survival and persistence advantage that allopolyploids have compared to autopolyploids. So it makes them sort of a interesting case for figuring out sort of these questions. So what am I doing this work in? I'm working in this wonderful genus of Clarkia. I saw some other Clarkiologists in the chat or in the room here, so they can appreciate just how awesome of a study system Clarkia is. For a variety of reasons, they're annuals. They are endemic to California. And just some fun notes about their natural history include the fact that they often have these specialist pollinators that only pollinate Clarkia. And they are, also good to grow because they're relatively easy to grow. They grow quickly. And as I said, they're annuals. So we can use them in experiments relatively easily. And sort of their natural history and ranges and ploidy and that sort of thing is all relatively well known. So the particular study, or like trio, I should say, of plants I'm focusing on are Clarkia stellata, Clarkia rhomboidea, and Clarkia virgata. Clarkia stellata and Clarkia virgata on the top and bottom here are two diploid progenitors of Clarkia rhomboidea, which is our allopolyploid here in the middle. And I've just mentioned this now, but this will also become more important later. Clarkia stellata is a selfer, and virgata is an outcrosser, and they sort of have floral traits that are sort of commensurate with that style of mating system. And if we look at sort of the geographic range of Clarkia rhomboidea, you can see that all these red dots are occurrence records, and it has greatly expanded its range sort of throughout California, as well as into sort of the Pacific Northwest, even into Idaho, and some populations in the mountains in Arizona and Utah, whereas stellata and virgata are sort of these narrow endemics in Northern California. And sort of importantly, the crux of my research really has been trying to develop this neopolyploid Clarkia rhomboidea, which I've successfully done by essentially crossing the diploids and treating them with colchicine, which is an agent that essentially affects the microtubule formation during mitosis, and it causes genome duplication, and you can generate neopolyploids in that way. So here's a recent flower of my neopolyploid Clarkia rhomboidea growing in the greenhouse, which is really exciting. So sort of the overview of what I'm going to talk about today, specifically with relation to Clarkia and polyploidy. I have three stories, one about reproductive isolation, secondly, sort of about local adaptation and range expansion, and third, about biogeography. And it sort of crosses like a scale of inquiry from very precise few populations to many populations and many species at large scales. So starting with reproductive isolation. So there's a kind of key problem for a newly formed polyploid, which is sort of presumed to be a major barrier to polyploid success, and that is called minority cytotype exclusion. And really this problem emerges from the fact that, a neopolyploid is going to be very rare initially when it first forms amongst its, from its diploid progenitors. And so you would predict under density dependent mating that the scale of pollen flow to the neopolyploid is going to really, overwhelm essentially its ability to either self-fertilize or, produce neopolyploid offspring. Or its pollen conversely will be wasted on trying to mate with diploids that it can not form fertile offspring with. But there's kind of two big problems with the assumption that minority cytotype exclusion actually is a huge problem for a sort of a neo-allo polyploid. The first is that there are a lot of pre-zygotic barriers that could limit the severity of minority exclusion. And the reason for that is because if pollen is not actually, flowing between diploids and polyploids, then minority exclusion isn't going to be happening in that way. And secondly, we don't actually know to what extent pre and to some extent post-zygotic barriers are present in neopolyploids. So what are some of these barriers? So there's pre-mating isolating barriers, including flowering, phenology, and pollinator isolation. There are post-mating, but yet pre-zygotic barriers, including the rate of pollen tube growth and competition of pollen tubes. And then there's post-zygotic barriers, including the ability for seeds to form. In this case, they would be triploid seeds, whether those triploid seeds are germinable, whether they can survive, and then whether those triploids produce any fertile pollen and ovules, or if they're totally sterile. So I'm going to talk about my experiments looking at pollinator isolation and some of these other post-zygotic barriers. So in terms of sort of the pre-zygotic component, do pollinator visits and pollen movement between diploids and neopolyploids reflect density dependence? And whether, in terms of the post-zygotic component, do triploid hybrids form, germinate, survive, and produce fertile pollen and ovules? So to get at the first question, I did this really big experiment last summer looking at pollinator visitation and pollen movement between inflorescences of the diploids and the polyploid. So what I did essentially is at two different sites, which were Blodgett Forest Research Station and the Institute for Forest Genetics, I set up these replicated arrays where I had 1, 4, 9, 15, and 18 inflorescences of Clarkia rhomboidea paired with the diploid found at that site. So at Blodgett Forest, the arrays were composed of rhomboidea and stellata. At Forest Genetics, the arrays were composed of virgata and rhomboidea. And I monitored sort of pollinator visitation and flight paths in these arrays. And I also used this really cool technique of labeling the pollen with fluorescent Q dots and collected the stigmas from every plant in every array. So essentially what that allows me to do is look at how much pollen from which species each individual received, which is pretty cool. So just to quickly show what this looks like, Blodgett Forest Research Station is kind of montane forest. You can see the array is set up here in the foreground. It's a little bit hard to see. And here's a bombus species visiting one of my flowers, one of my rhomboidea flowers. And then this is what the Institute for Forest Genetics looks like. It's more of a low elevation site with kind of oak and pine woodland as sort of the primary habitat type. So we're going to look at some data here now. So on the x-axis is the frequency of C. rhomboidea in the arrays. And then on the y-axis, we have the number of total number of visits to the array, with the diploid sulfur here on the left and the diploid outcrosser here on the right. So when paired with the sulfur, you can see that the total sort of number of visits to the arrays increases as rhomboidea increases in frequency, whereas there's kind of a general decrease in visits when it's paired with the diploid outcrosser. In terms of the length of these visits, so again, we have polyploid frequency and visit length on the y-axis. When paired with the sulfur, we can see that visits to the arrays tend to get longer. Pollinators spend more time in the arrays as rhomboidea frequency increases, the polyploid. And we see the opposite when rhomboidea frequency increases when paired with the diploid. So pollinators are spending less time in the array when rhomboidea is more available. Now we're going to look at not just like visits overall, but visits in terms of pollinator flights. So this is actually looking at not only like what species did a pollinator visit, but where did it go after it made that visit. So these flights, I'm calling them, are sort of classified in three different ways. They can either be within a single species, so either the pollinator moves within an inflorescence or moves between inflorescences of the same species, or the pollinator can move between inflorescences of different species. So those are the kind of categories we have here. And so again, if we take a look at our axes, we have the array proportion of C. rhomboidea, the polyploid, and on the y-axis, we have the proportion of different kinds of pollinator transitions. Red will be between, green will be within the diploid, and blue will be within the polyploid. And these dashed lines represent our expectations under density-dependent mating. So like if the bees had just come to the array randomly, what would the data look like? This is what it would look like. And then this is what we actually see. So the first thing to notice here is that there's sort of disproportionate visits to the polyploid throughout the different arrays of different frequencies. So even as rhomboidea frequency increases, the bees consistently stay sort of visiting rhomboidea, the polyploid. And then the other sort of key thing here is if you look at the red line, there's fewer transitions, fewer flights between the two species than expected under density-dependent mating. Okay, so now we're looking at paired with the diploid outcrosser. So this is rhomboidea, the polyploid, with the diploid outcrosser. We have the same sort of setup as far as our axes and our expectations. And what data or what results do we see? What do we find? Okay, so we see that essentially we get the opposite result, where instead of having disproportionate visits to the polyploid, we actually have disproportionate sort of visits that are staying within the diploid. So most bees are just happy to stay visiting Clarkia virgata, even though it becomes quite uncommon in the array. And in fact, this last point here, where polyploid visits exceed sort of the diploid ones, this is only one virgata in this array and 18 rhomboidea. So there's like a huge difference between them, and still the diploid gets lots of visits. And there's, again, this same pattern emerges here where there's a lot fewer flights between the two species than expected. So it's pretty clear that there's not, random movement among these inflorescences by the pollinators. So what are the sort of key takeaways here? When virgata and rhomboidea are together, remember rhomboidea is the polyploid, we see fewer flights between the polyploids and diploids than expected. Fewer flights between the polyploids than expected, and fewer interspecific flights than expected. And when paired with stellata, we see essentially the opposite, where more flights are staying sort of within the polyploid, but again, we see fewer interspecific flights than expected. So sort of forthcoming from this, I still have to analyze the pollen movement data and see whether or not that sort of accords with what we see in terms of pollinator visitation. And sort of the longer term goal is to essentially repeat this experiment with the neopolyploid and find whether or not the lack of evidence for density dependent movement is also true when I put arrays of the neopolyploid with the diploids, and whether or not those results sort of diverge from the natural C. rhomboidea. But sort of overall, these results are pretty suggestive of the fact that it doesn't seem that, density-dependent sort of movement is occurring. There is definitely pollinator sort of discrimination of these different species, even though they do look quite similar. Okay, so moving on to the post-zygotic component. Do triploid hybrids form, germinate, survive, and produce fertile pollen and ovules? So if we're looking at this, one important note is sort of that endosperm can play an important role in polyploid hybridization success. And this is really reflected in its impact on sort of the formation of seeds and whether or not they can germinate. And there's sort of two related factors here that are related concepts, which can provide us with a useful framework for making predictions about how successful crosses between polyploids and the progenitors will actually be. And those are endosperm balance and parental conflict. So I'll just kind of briefly overview these concepts. So The key thing to know here is that endosperm balance is upset in diploid polyploid crosses. And the way that works essentially is when you have diploids and a polyploid, they have different copy numbers of their genome, right? So Virgata has 2 Virgata genomes, Stellata has 2 Stellata genomes, Rhomboidea has 2 each, 2 Virgata and 2 Stellata genomes. If we look at the embryo and endosperm and pollen of these species, we'll see what those constituent parts are. And I should mention here that Clarkia has diploid, not triploid endosperm. So that's a bit of a wrench in your normal thinking about this sort of thing. But just remember that Clarkia has diploid, not triploid endosperm. And so the way this works out, basically, if you were to make crosses, for example, is that if Rhomboidea is the father in a cross with one of the diploids, it will bring a complement of the V and S genome along to the endosperm of the diploid parent. And if it's the maternal parent, the endosperm will have one or the other, or sorry, will have both complements of the genome. So when you make these crosses, you end up with these different ratios of maternal to paternal genome copies in the endosperm. So you end up with 1 maternal copy and 2 paternal copies when Rhomboidea is paternal, and the opposite case when Rhomboidea is maternal. And so, as I said, endosperm balance is upset in these diploid polyploid crosses. The key thing, what I mean when I say upset, is that we want sort of the normal ratio. So in Clarkia, the normal ratio is 1:1, whereas here we end up with 2:1 and 1:2 ratios depending on the cross. So for example, Rhomboidea by Stellata, we end up with a 2:1 maternal to paternal copies in the endosperm. And that's a maternal sort of deviation from normal. Or if we did a Stellata times Rhomboidea cross, we end up with a 1:2 ratio of maternal to paternal copies. And that's a deviation towards that favors sort of the paternal genome. And sort of we can make predictions about how these things will influence the viability of seeds. Typically, maternal bias leads to greater viability in smaller seeds, whereas paternal biases leads to lower viability and larger seeds. Okay, so I know that's a bit of a lot. Now we're going to add one more layer to this, this scheme here called parental conflict. So the idea here is that mating system, or the mating system of the species in question, can actually influence cross success via parental conflict. So what is parental conflict? So if you consider 2 species that differ in mating system, here we have a selfer called Stellata and an outcrosser called Virgata. They may receive, if you're a selfer, on the stigma, pollen from one source. Whereas if they are an outcrosser, they receive pollen from potentially many pollen donors, many potential fathers. So you end up with ovules in the case of a selfer that are all fertilized by a single father, whereas in the outcrosser, you have many fathers. So this results in differences in the competition amongst individuals for maternal endosperm allocation, and long-term leads to differences in selection for competitive alleles. So in outcrossers, you predict there's this highly competitive environment for endosperm allocation, and ultimately the father is trying to override the mother in terms of getting endosperm resources, whereas that's not occurring as much in the selfer. So this has consequences for what happens when you cross species that have different mating systems. If you make a selfer maternal cross to an outcrosser paternal cross, you end up with endosperm overgrowth because the father's pollen and gametes contain alleles that promote essentially highly competitive resource allocation from the to the endosperm. And in the reverse case, when you make maternal outcrossers cross with selfers, you get suppression of the endosperm. So this is all parental conflict. And this parental conflict can sort of map onto our endosperm balance idea. So, if we have these maternal versus paternal copies of endosperm, or genome copies in the endosperm. And we just sort of arbitrarily say that because Virgata is an outcrosser, it gets a higher value for its genome than Stellata does. We can sort of adjust our endosperm ratios accordingly. So the 2-to-1 bias for the Rhomboidea x Stellata cross becomes, for example, a 3-to-1 bias because Rhomboidea carries the Virgata genome as well as the Stellata genome. And we can do that for all of our crosses, and we can see something interesting emerge here, which is that the crosses involving Virgata are closer to 1. They're closer to being balanced than the crosses involving Stellata. And so we can guess that these will be more viable crosses, these crosses involving Clarkia rhomboidea. And we can also predict that the maternal rhomboidea cross will be more successful than the paternal cross because it has the bias favoring it in that direction. And the maternal crosses usually are better. So what do we see, or what do I do to actually test all this? So I did some experimental crossing where I did reciprocal crosses with many populations of the diploid and the both diploids and the polyploid. And I got the mass of the unaborted seeds and the seed viability with a germination assay. So if we look at the results in terms of viable seeds per fruit, that's what we have on the y-axis here. And on the x-axis, we have our crosses. So the first 2 crosses are the 2 rhomboidea polyploid maternal crosses. And then the second 2 are the rhomboidea paternal crosses. What do we see? We see that the 2 crosses involving Virgata, the diploid Virgata, so RV and VR, have much greater viability than either cross involving Stellata, the other diploid, the sulfur. So our predictions do hold. We find that the Virgata crosses are more viable than the crosses with Stellata. And we find that the maternal rhomboidea x paternal Virgata cross, RV, is more viable than the reverse cross, VR. There is a bit of a sort of a complication here, though, which is that I also found that in maternal rhomboidea crosses, there's a proxy for outcrossing, or a proxy for outcrossing is associated with decline in hybrid seed mass. So what does all that mean? So basically, this complicating factor here is that we know that Clarkia rhomboidea populations could have variable histories of outcrossing, which could create variation in parental conflict. And this is sort of like explained or was documented when like rhomboidea was first sort of taxonomically explored back in the 1960s. But essentially, the point here is that it seems clear that rhomboidea populations do vary quite a bit in their extent to which they self versus outcross. And so I thought that that could have an influence on whether or not these crosses are successful because of this parental conflict issue. So when a rhomboidea population is more outcrossing, which is what we have on the x-axis here, how does that influence hybrid seed mass when crossed with either diploid? We find a negative trend here, which suggests that there is, in fact, an influence of the history of outcrossing in rhomboidea populations on essentially the hybrid seed mass. So what's happening down here at this end of the spectrum, at the sort of far right side of the graph, would be that individuals from those populations have sort of more competitive alleles that can resist paternal endosperm overgrowth. Whereas when they have sort of low amounts of outcrossing and higher autofertility, they don't have those same competitive alleles, and hence the hybrid seeds are heavier because the paternal overgrowth is not resisted. Okay. So if minority sort of exclusion is acting on C. rhomboidea, I think it will involve sort of triploid fertility because, as I've shown you, there is actually a great deal of pollinator isolation between these species. And as I just sort of showed you, triploid seeds apparently form quite readily. They germinate fairly well. And I'm actually growing them all in the greenhouse right now. All of the offspring from those crosses are currently growing and surviving so far. So sort of the key point here will be understanding how fertile those individuals are. Whether they have fertile pollen and ovules will be key for sort of getting at whether or not more broadly minority exclusion could be expected to act on C. rhomboidea. But so far, the data suggests that pollinators really reduce the chances of hybridization. And if it does in fact occur, the hybridization may actually prove to be far less deleterious than expected. But again, it really hinges on how fertile sort of the triploids turn out to be. Okay, so moving on from reproductive isolation, let's talk about polyploidy per se and sort of later versus later adaptation. So polyploidy per se or does polyploidy per se or later adaptation influence adaptation to novel environments? So there's kind of two parts to this. One is a habitat shift and two is a range shift. And I'll kind of talk about these both in part. Oh, my office light is turning off by itself. Sitting too still. So the first thing to understand here is sort of the differences in habitat between the diploids and the polyploid. So the diploid progenitors of Rhomboidea, they tend to be in either open slopes and sort of woodland areas or like openings in montane and alpine forest, sort of characterized by like disturbance and highlight and higher competition. Whereas the allopolyploid, Clarkia rhomboidea, it sort of lives in the interior of montane forests. And some big differences include the fact that this is a relatively low light environment. There tends to be a lot of snow cover in these environments. It lasts longer, and there's a lot less competition in these environments. So this picture kind of shows you like this is typical Clarkia habitat. This is a site that has lots of Clarkia virgata at it. And then this is sort of the typical Clarkia rhomboidea habitat. You can see that, we're inside of a forest, we're on the forest floor, it's quite shady, and there's not a whole lot of competition. Everything here that you can see really in the foreground of both these pictures is Clarkia rhomboidea. There's not a whole lot of other species growing in sort of this understory environment. Okay, but what is the role of polyploidy per se in this shift? So when I say polyploidy per se, again, I'm really talking about the increase in cell size and its sort of downstream consequences on physiology and niche. So when moving from the diploid to the neopolyploid to the natural polyploid, sort of what is the contribution of polyploidy per se in terms of this habitat shift? And then sort of secondly, what is the connection to the range shift? So we could conceptualize, for example, a neopolyploid has formed. It has some sort of alternative habitat or niche than the diploids do, and that habitat or niche is widespread, and so therefore the neopolyploid becomes widespread. Sort of conversely, we could think about a situation where the neopolyploid, is different from its diploid progenitors in some way, but then range expansion has occurred via adaptation. So it's not that there's some sort of common environment that's really common around globally. It's just that the neopolyploid, for various reasons, has a strong ability to adapt to sort of a bunch of alternative environments. And in both of these cases, you would expect range expansion. But there's sort of an underlying challenge here in that when we're talking about allopolyploids, we're talking about both hybridization and polyploidy. So when you're looking at sort of a neopolyploid, it could be unclear sort of what contributes to some factor you're interested in, like habitat or range shifts. You could be observing the impacts of hybridization, or you could be looking at the impacts of polyploidy per se. So you have to have essentially both of these in your experiments, the hybrid and the neopolyploid. Okay, so to actually sort of answer this question, what do I need to show exactly? Or what do I aim to show? So essentially, I'm going to talk about a field experiment that I'm setting up to get at sort of three different things that I need to show here. One is that differences in range among the species are not due to dispersal limitation. So basically confirming that the fact that these species live in different sites is not just because they haven't dispersed there. The second is that I need to show that polyploidy per se sort of improves fitness over the F1 hybrid in the polyploid habitat. So whether or not sort of the neopolyploid is actually capable of living in those interior montane forests where Clarkia rhomboidea lives. And then also I would need to show that there's not evidence for sort of local adaptation of C. rhomboidea if I wanted to show that sort of alternative niche happens to be widespread, and that is sort of the driving force behind range expansion. So the field experiment is set up like this. Basically, I have 3 source populations of each of the 3 Clarkia species, the 2 diploids and the polyploid. And then I have the diploid F1 between them, involving sort of multiple cross directions and the source populations. And then I also have 4 different lines of the neopolyploid that I got from the diploid F1. So all sort of 5 groups here are going into this field experiment. And I'm actually currently working on standardizing their maternal environment with a greenhouse generation. And the field experiment will be sort of set up in this way. I'll have 9 source populations, so 9 transplant sites. And then at each transplant site, there will be these arranged blocks of transplant planting grids. And then individuals will be randomized into these grids, individual seeds. And then data analysis will look something like this. So we use sort of like a life history-based fitness analysis that takes into account every stage of the life of a plant. And really the sort of advantage of this is that when there are differences in fitness, you can sort of pinpoint whether those are being driven by, for example, differences in germination at a site or differences in, say, survival at a site, et cetera. And all of this is incorporated into sort of an estimate of lambda, which you can think of as the seeds per seed input into a transplant. And sort of the potential results here. So, if it is true that polyploidy per se is the main driver of this shift in habitat, then the evidence that I would need would be that the fitness of the neopolyploid is sort of greater, the F1 hybrid, and similar to the polyploid in polyploid sites. If that is not true, if there's kind of a small effect of polyploidy per se or no effect, then I would expect that the fitness of the neopolyploid would be similar to or less than the F1 hybrid when planted in polyploid sites. And then in terms of range shifts, so if it is true that, the polyploid is sort of adapted to an alternative habitat or niche that is widespread, then evidence in favor of that would be that the fitness of the natural polyploid is sort of stable across interspecific sites, more so than diploids. And then alternatively, if in fact range expansion is driven by adaptation, then we would expect, greater fitness planted at home sites than elsewhere and greater fitness at home sites than foreign populations when transplanted to that site. So essentially evidence for local adaptation. So the significance of these results would include like providing direct evidence for the contribution of polyploidy per se versus hybridization and later adaptation to observed habitat and range shifts, and then also sort of uncovering contributing mechanisms to polyploid range shifts and range expansion. And just for fun, I have some pictures documenting the challenges of this kind of field work. So some of these challenges are like part of the interactions and part of the things that we actually want to measure, like herbivory. You can see this poor seedling got chewed down, or humans, which are not the kind of biotic interaction we're hoping to measure, but nonetheless, they can tear up grids and do other things. Also things like competition, again, that's sort of part of the story, and snowstorms burying or seedlings in 2 feet of snow in the middle of March. What can you do? You can't really do anything about it. But again, these are sort of the things that we want that are, inherent to the environment at those sites. And that's why we use high replicate numbers. Okay, so the last story is about biogeography of polyploidy and Clarkia. So there's been this pretty persistent idea in the literature, generally speaking, that polyploidy drives range size increases. And you'll find that this is often cited, Clarkia is often cited as an example of this. There's a relatively well-cited paper from 2006 that suggests this to be the case. That's where this figure comes from. And the sort of hypothesis here is that polyploids must have some kind of adaptive advantage over diploids to consistently achieve these larger ranges. But, some among you may be wondering, well, what about, is this corrected for phylogeny? The answer is no. but I'll sort of get into how we can kind of do that by looking at comparisons to specific diploid progenitors. And also, sort of more broadly, I think I'm going to start digging into this alternative hypothesis for why they could have different range sizes, larger or smaller. And that is this idea that polyploidy per se drives some sort of common niche shift in polyploids that may lead to smaller or larger ranges. Okay, so first, I'm going to just step back and articulate a few questions that get at these hypotheses in various ways. And I'll kind of explain how these questions relate to those hypotheses as we move forward. So the first one here is, do polyploid clarkia have actually larger geographic ranges than diploids, and specifically diploid progenitors? So if there is an adaptive advantage to polyploidy, then we would sort of expect that polyploid ranges would routinely be larger than the progenitors. If this common niche shift is actually the explanation, then we would not predict that polyploid ranges would be related to the range size of their progenitors. So in order to do this, I gathered occurrence records of all clarkia and their ploidy. I removed geographic errors, and I calculated their range size with this RCA method, which essentially involves placing 25-kilometer diameter circles over every occurrence for each species and dissolving the overlap. Okay, so we're going to look at some results now. So on the left here, you have the latitudinal range, and on the right, you have the range size of the diploids and polyploids, diploids in red, polyploids in blue. And we can see that although polyploids have this larger average range size and latitudinal range, you can also clearly see that this pattern is driven by small sample size and some key outliers. So there's only 10 polyploids in Clarkia. Well, I say only, but there's 40 diploids, 30 diploids here, 30 some diploids, and 10 polyploids. So it seems sort of premature to suggest, based essentially on just these two outliers, rhomboidea and purpurea, that polyploidy is like clearly driving an increase in range size. Obviously, these two polyploids have large ranges, but that doesn't seem to justify just saying, oh, well, polyploidy drives range size increases and stop there. So where do we go from here, if that's the case? We can look at progenitor polyploid pairs. So polyploid ranges are both larger and smaller than their largest range diploid progenitor. So again, we have range size on the y-axis, the diploid progenitor on the left, and the polyploid on the right. And you can see that in 5 cases here, the polyploid actually has a smaller range than the larger ranged of its 2 diploid progenitors. And then only in 2 cases do we see like huge increases in range size. So again, it's really just reinforcing this idea that it doesn't seem clear that polyploid ranges in Clarkia are routinely larger than their progenitors. And they seem more or less unrelated to the range size of their progenitors. So really no evidence for this sort of adaptive advantage hypothesis. Okay, so the second question is related to climatic rarity. So does climatic rarity sort of set an upper boundary on range size, and does it predict polyploid occurrence? So sort of the key idea here is that a given environment can be rare or common in a landscape, and we find that rare climates are more likely to host small range species. It's a fairly well-established phenomenon from this paper, Olemüller et al. 2008. So how do we like conceptualize what this would look like for Clarkia in this scenario? So if we're looking at rare to common climates on the x-axis here, some measure of how common a climate is, we would expect that there would be sort of this boundary effect on range size, where range size couldn't be very large in rare climates, but range size could be both small and large when climates are more common, or when a set of climates is more common. And then in addition, we also expect that as climates become more common, we'll see a sort of increase in this polyploid fraction. So how do we calculate climatic rarity real quick? So for a set of occurrences, so these are just some this is the occurrences of all Clarkia here on the left. For a given location, we can sample the environment that is present in that location, or the set of environments. We can obtain what's called a multivariate environmental similarity surface, and essentially calculate the size of it. So basically, what this is telling you is the geographic area of similarity in relation to the focal environment that you sampled initially. And so I did this in two ways. I did this by sampling sort of random boxes of occurrence records from within the range of individual species, and also by sampling random boxes of all species records where the polyploid fraction was variable, so across the whole range of all Clarkia. So with this first method, if we're looking at sort of the x-axis here, we have our climatically similar area. On the y-axis, we have range size. And we find that Clarkia species do in fact show this evidence of this sort of boundary effect, where they do not inhabit both large ranges and rare environments. So you can see where there's rare environments, there are no large range species, but as the environments become more common, we see greater variation. Some species can have large ranges, some can have small ranges. And it seems to be, this effect seems to be driven in part, at least by these polyploids that have large ranges. And then we can also look and see that areas of high polyploid fraction are environments that are common in the landscape. So this map on the left sort of shows in orange, the more orange it is, the more polyploids occur there. So polyploids are really common sort of Northern California into Oregon and Idaho, and they're much less common sort of in the coastal areas and Southern California and the sort of foothills of the Sierra. And we can look and see, again, here on the x-axis, we have climatically similar area. On the y-axis, we have polyploid fraction. We can see that there's a nice association between climatically similar area and polyploid fraction. So essentially areas where the climate is very sort of common and homogeneous over wide areas tends to host a greater proportion of polyploid Clarkia compared to diploid Clarkia. So again, it doesn't really seem that we have evidence for the adaptive advantage that is purportedly associated with Clarkia polyploids. Okay, so the last part of this is whether or not our shifts in range are a predictable outcome of niche shifts that accompany polyploidy per se. So I already mentioned that it's pretty clear that there's not really strong evidence for polyploids having larger ranges in Clarkia, but they do have different ranges. And so what is underlying sort of these differences in their range compared to diploids? So again, when diploids transition to neopolyploids, there are these cell size effects driven by polyploidy per se. Some of these include things like increased nutrient demands, increased secondary metabolite production, increased seed size, decreased max metabolic rates, decreased growth rate, et cetera. And these are sort of presumed to do things like decrease the flexibility in the set of habitable environments compared to diploids, and sort of downstream leads to sort of potentially greater homogeneity of environments in which a polyploid can exist compared to a diploid. So in terms of our hypotheses, we would predict that if that's not true, if in fact polyploids have some sort of adaptive advantage, then we would expect that environments in part of a polyploid's range cannot actually predict the entire range of that species, because has been able to adapt to a diverse set of environments. Whereas if this sort of common niche shift hypothesis is true, we would expect that environments in part of a polyploid's range can successfully predict the entire range. All right, so just to back up a second and talk about sort of the broad scale differences between diploid and polyploid ranges and the sort of environments within them. Here we have this principal component analysis that's just based on sort of a quick overview of what environments diploids and polyploids tend to inhabit. And we can see that they really separate along these axes involving things like temperature seasonality and precipitation seasonality, where polyploids are in environments that are much more seasonal in temperature, but much less seasonal in precipitation. And also, interestingly, more historically unstable in terms of climate, like things like glacial fluctuations. So we use species distribution modeling to test whether polyploid ranges are in fact more predictable than diploids. The way we did that is by taking species records and climatic variables, clustering them using k-means clustering, building species distribution models using Maxent, which is a common SDM algorithm, with only those clusters, and then testing those models on the full range of the species in question. And then we evaluated those models with TSS, which is this model performance metric, essentially asking how well does an environmental cluster of records predict the range of the entire range of the species. So high TSS would sort of give us an indication that the environment is homogeneous across the range. Low TSS would give us evidence that the environment is more heterogeneous across the range of that species. Okay, so we're going to look at these results in terms of our diploid progenitor and polyploid pairs. And on the x-axis, again, we have ploidy. On the y-axis, we have TSS. And I'm just showing you the results from when k-means clustering was done at k equals 7, which is basically the most restrictive scenario, or most conservative scenario, because we have the smallest sets of clusters in this case. And this is what we find. So when going from diploid to polyploid, we find that the polyploids in general are more predictable from subsets of their range than the diploids, with the exception of this one case here, the Amina gracilis pair. So what this really seems to show, with an exception, is that polyploid ranges are more predictable from a very narrow environmental subset of their whole range compared to diploids. So again, this does not really support the adaptive advantage hypothesis, because diploids seem to inhabit more varied environments, even though their ranges may often be smaller than some polyploids. I guess I should mention, even in the case of Stellata rhomboidea here, Clarkia rhomboidea has a much, much larger range than Clarkia stellata. It's the largest range of all the Clarkia, and yet its range is more predictable from a small subset of the environments in which it inhabits compared to Clarkia stellata. So just to overview these findings, polyploidy is not consistently associated with range expansion in Clarkia. Climatic rarity does seem to set sort of an upper bound on range size and does predict polyploid occurrence. And polyploid Clarkia ranges tend to be more environmentally homogeneous. So altogether, these results kind of suggest that shifts in range for polyploids represent sort of a realization of an alternative niche, which may be constrained in part by the effects of cell size and by the availability of the niche on the landscape. So just to sort of wrap up all of that, I threw a bunch of stuff out really fast, but my idea here is to sort of really illustrate and connect sort of the early stages of polyploid formation through to whether or not they can sort of persist once they formed because of minority exclusion, whether the scale of sort of the niche shift that occurs in polyploids, and then the downstream consequences for geographic range. So with that, I just would like to acknowledge my wonderful lab and the many generous funding sources, and especially the land managers in California who host my many experiments, including Blodgett Forest Research Station and the El Dorado National Forest and various other U.S. Forest Service entities that have helped me a lot, including the Institute for Forest Genetics has been a huge help for my research. So with that, I will take questions.
All right, great. Thank you so much, Aidan, for that talk. Learned a lot about polyploidy in the process that I didn't know before. So thank you so much. And we're a little over 1 o'clock, but if anyone would like to ask a question, maybe we'll have time for one or two.
Sure.
Yeah, you guys can type in the chat and I'll give you a minute, or unmute yourself. And I can just start out if we only have time for to get through it quickly. I was wondering like how difficult is it to make the neopolyploid? And I asked that, and maybe it is really difficult, but I asked that because like the polyploids were variable in some of their characteristics, as you showed with the range, for example. And so when you're comparing the neopolyploid you generated with a wild polyploid, do you need like a bunch of neopolyploids to say something?
It is true that there is possibly large variation sort of between neopolyploid lines. Um, and yes, it is difficult to create them. So
Mm,
I do have 4 lines.
mm.
So it sort of remains to be seen whether or not, those are really different from each other or similar.
Yeah.
Um,
Did
yeah,
you when you made your four, did you try to use diverse samples for
yeah, they involve different populations.
got you. Very cool. Well, excited to see what comes of it. Thanks so much again. I think I'll end it here because of the time, but really want to thank Aidan, and we'll see everybody next week for the next Greg seminar. Thanks so much.
Thank you.